Thank you for having me. My name is Lauren Seyler. I'm a postdoc in Matt Schrank's lab at Michigan State University, and I've been working on metabolomics as a means of mapping metabolic networks in microbial cultures and also in environmental samples since I started in this lab about 1.5 years ago. And so today I'm going to talk to you about a particular site that I've been working at, the Coast Range ophiolite in California. Oh look, it's the same slide that Katrina had. It's almost like we worked in the same lab. So you've already seen this, you're already familiar with the reactions that are associated with serpentinization. But what I really want to draw your attention to is the part of this reaction that interests me the most, and that's this production of methane and these small organics. And what's interesting about that to me is that while microbes can utilize methane and these small organics that are produced by this reaction, they're also producing these things themselves. We know that microbes can produce methane. They produce these small organics, and so it creates this really amazing and confounding gray area between geochemistry and biochemistry, which I think is really at the crux of not only astrobiology, but origin of life questions as well. And it creates this beautiful, loving relationship between microbes and rock. And so, as I mentioned, I'm working specifically at the Coast Range ophiolite. It's just one of many serpentinizing environments that our lab is studying. And this site is about 3 hours north of San Francisco, or an hour north of Napa. It's a really nice drive up the coast, and stop through wine country. And there's two particular sites at the Coast Range ophiolite Microbial Observatory, which is shortened as CHROMO, and that's Quarry Valley and the Coreshed Well. And each one of those sites has a cluster of wells. There's 3 at Quarry Valley, and then there's another 5 plus an older well that was drilled before this project at Coreshed Well. And this is what the wells look like. Not very exciting to look at on the surface. And I took these samples in May and June of last year. And this is some work that Katrina did while she was still a PhD student in Matt's lab, looking at 16S diversity in some of these wells. And what I want to point out, this is a figure from her latest paper. What I want to point out here is the incredible decrease in alpha diversity that occurs as you go from these lower pH wells like CSW14 down to these very high alkaline wells like QV11 and CSW11. So as you get into these higher alkaline wells, these deeper wells, we really see a decrease in the amount of diversity that we see, and these communities become very, very simple. And that's why I decided to focus on these 1,1 wells, because they are hyperalkaline and they are very simple communities, and I thought that they best reflected this relationship between microbes and the serpentinizing system. And so if we just want to look at the 1,1 wells at a glance, these are the deepest wells at these sites, with the exception of CSW old, and they are the highest pH. As I mentioned, they're the least diverse, and so there you have the simplest communities. They're also uncased, which means that they can be influenced by water coming from the surface, like rainwater, which there isn't much of in this area, but it is a concern. And so we can have this percolation of organic material from the surface down into this well water. And if we just look really quickly at some of the chemical parameters of these wells, they're about the same depth. The pH ranges from 11.5 to 12.5, highly reducing, very low dissolved oxygen, and relatively low biomass compared to something like seawater, for example, or surface streams in the area. But again, I want to draw your attention to the concentration of methane in these wells and some of these simple organic acids like acetate and formate. In the QV wells in particular, these organic acids are sometimes below detection limits, but that doesn't necessarily mean that they're not being produced. They may just be cycled very, very rapidly. So we're not really sure if the microbes are producing methane, or if they're consuming methane, or if they're producing something else entirely. If we look at the metagenomic data, which was provided by Billy Brazelton, who's now a professor at University of Utah, we see a lot of pathways for things that we would expect in a well that's influenced by groundwater percolating down into the wells. So genes associated with the hydrolysis of cellulose from plants, for example, or the degradation of polyaromatic hydrocarbons, fatty acid catabolism. We also see a ton of gene pathways associated with fermentation, which makes sense because it's a very low oxygen environment. So the production of acetone, for example, which fermentative cells use to store excess acetate. And this makes sense when we look again at the chemical parameters of the wells. We can see this production of formate and acetate through all of these fermentative processes. So that's interesting, but what about the methane? Well, if we start looking at the genes associated with methane production and consumption, we start to get a really interesting story here. So the reductive acetyl-CoA pathway, or the Wood-Longjaw pathway, isn't really prevalent in these wells. And neither are the genes for methanogenesis, which makes sense because we weren't really able to find any evidence of methanogens in the 16S data either that comes from these metagenomes. So it doesn't really look like methanogenesis is happening in these wells. There is methanogenesis from acetate here in a small amount, but if we can't find the methanogens, then that could be there because of horizontal gene transfer, or maybe it's been mislabeled, any number of things. We do find quite a bit of acetate formation from acetyl-CoA. And then all of these available genes and gene pathways for the consumption of methane for methylotrophy. And not quite as much methane oxidation to CO2 as I would have expected. So there's all of these potential pathways for consuming methane and using it up, but not necessarily pushing it to CO2. And perhaps that's because CO2 precipitates out so readily in these systems. I'm not really sure. So we have this very complex metabolic network here that we're trying to tease apart. And while we have all this metagenomic data, all that's really telling us is that these cells have the potential, or they have the knowledge for these processes, not necessarily that these processes are actively occurring. And that's where metabolomics comes into play. So for those of you in the audience who are not biochemists or not microbial ecologists like me, a metabolite is a small organic molecule. It's a product or an intermediate of a metabolic process. And the metabolome is a collection of all the metabolites produced by an organism, a population, or a community. So metabolomics is really a snapshot of metabolism as it's occurring. We have genomics, which will tell us that a cell has the knowledge of a process. Proteomics will tell us that the cell's producing the tools to perform that process. But metabolomics is evidence of the process in action. Of course, there are some limitations. And as I mentioned before, there's not a lot of biomass in these wells. So in order to really get at some of the rarer metabolites that are like the smoking gun of metabolism, we need quite a bit of carbon on these filters. If we assume that each cell contains about 30 femtograms of carbon, that means I would need to filter about 50 liters of water. That's a huge amount. And while the 1-1 wells have a pretty high fluid output, that's not true for all of the wells at this site. And there's also limitations just to the filtration. The filters can clog because as this highly alkaline water comes in contact with the atmosphere, lots of carbonates precipitate out, clog the filter. It's difficult to keep this water cold to prevent all kinds of metabolic interactions from occurring as you're filtering. So the data shown here is for about 12 liters of water. So it's really a two-step process. First, I filter the water through vacuum filtration in a glass stand like this through a PTFE filter. And then, yes, that's the front seat of a car. This is the best picture I had of me actually doing this in the field. And then that filtered water is run again through an SPE cartridge, which stands for solid phase extraction, which captures all of the dissolved carbon in the sample so that can be run separately. So we're looking at two pools of metabolites here. Whatever's trapped on the filter, which represents intracellular metabolites, and then all of the dissolved organic matter that's present in the well water. And if we look at that and compare those samples on a PCA plot, we see that the fluid samples are sort of similar to each other, but drastically different from everything else. What's happening? It's not going over. There it goes, sorry. And then the biomass samples are very, very similar to each other. And there's our blank and our biomass blank. So if we look at all of the different compounds that we've observed, which are analyzed on tandem liquid chromatography mass spec, we had a total of about 6,800 compounds. When we subtracted out whatever ones were also found in the blanks, that was about 4,500. And only 79 of those samples, or those compounds, were found across all of the samples. So there were a lot of compounds that were very unique to the extracellular fluid, and only a few that were unique to the biomass. And the CSW well had a lot more unique compounds than the QV well for whatever reason. There's also a ton of possible identifications for all of these compounds. We search the database and compare with the mass to charge ratio, retention time, fragmentation, and try to figure out what all of these compounds are. And there were 50,000 possible IDs in the human metabolome database, which we were able to narrow down to about 3,600 in the KEGG database. So some of the common compounds that we found here are some compounds of interest. There are lots of different vitamins, including a lot of B vitamins, and the metabolites of those vitamins. Quinones, which are used in respiration and for different metabolic processes. Aromatic hydrocarbons, acetone acetate, which I mentioned is linked to fermentative processes, and antibiotics and the products of the degradation of antibiotics. All very interesting, but what was most interesting to me is that I found all of the compounds that are associated with this RUMP formaldehyde fixation pathway associated with methylotrophy. All of the compounds associated with the serine pathway for formaldehyde fixation. Some of the ones associated with tetrahydrofolate pathway, and some of the ones that were associated with the glutathione pathway as well. So there's plenty of evidence here for methylotrophy occurring in these wells, not only in the metagenome, but also in the metabolome. And very little evidence, I believe, for methanogenesis, at least in this particular site. We still need to use some more sensitive methods to measure some of these small intermediates because LCMS doesn't really pick up the very small metabolites, especially uncharged ones. And we have a ton of unidentified compounds that may also be used as possible biomarkers for looking for microbes associated with serpentinizing environments, potentially on other worlds. And they may be very, very useful for those kinds of studies. So I'm running out of time, but the next steps that we're going to do in this project is we're going to construct metabolic networks in R using the metabolome and the metagenome. I'm going to do some further work in chromo, including some work at CSW old because it is such a deep well and it is so interesting. It's got very low amount of biomass, but I think there's a lot of potential there. I've also been using these same techniques in the Oman drilling project, and the picture from the front seat of a car was from that project. That's an entirely separate funny story. I'm also using these techniques on microcosms and enrichments in the laboratory. We're talking about potentially using them on microcosms from Lost City. And definitely, if you're interested in this work, and particularly the work that we're doing at Chromo, you should check out Mary Sabuta's and Lindsay Williams' talk this afternoon in this room. And hopefully, we'll be able to use some of this work to eventually find a microbe in space. And so with that, I'd like to thank you for your attention, and I welcome your questions if there's time.
We have time for a question or two for Lauren. Oh, yeah, in the back, if you could either speak loudly or come up to a microphone.
So I'm very interested in your enrichment experiments because oftentimes in the lab we see lots of diversification happening. And I kind of wonder if that's the thing you might expect in your system, or is it going to stay possibly more simple like some of the other speakers have discussed?
So I'm still working very preliminarily on some of these enrichment experiments and microcosms, but if you're interested in enrichment experiments from this site in particular, definitely check out Mary's talk this afternoon. But we've been basically providing a reductant to these microcosms and seeing if we can observe methylotrophy in the production of CO2 in our lab.
Hi, Jamie Foster, University of Florida. I'm really curious about the metabolomics. Did you see any cycling, any diel cycling within your community, or is this just a single time point? What's the reproducibility of your signatures in your community?
So this is a single time point. I initially took triplicate samples, but the signal was so low that I had to pool all of them, and that's why it was a 12-liter sample, because originally it was 4. In samples that I've done in culturing, these results are very reproducible. Triplicate samples look very, very alike. As far as reproducibility at this particular site, this is still very preliminary, and we're still working through the methodology, and so I was only able to take one sample from each well. But we'll be going back and hopefully doing this at a larger scale in order to show that reproducibility and maybe see some differences seasonally or with other parameters.
All right. Any other quick questions? All right. Well, thank you again, Lauren.
Thank you.